actually after about a year and a half in the lab, I think. So Noam started as a computer science, computer engineering.
Yeah.
Computer science.
Computer science.
Right. So he was a computer science student, and somehow he did a project with me, and then I think what happened actually historically is the first you attended the course about learning and memory.
Yeah.
Which, thinking about it, was given more than 2 years ago, so that's quite a while. And then you started working in the lab, and I think the work slowly evolved and turned out to be very nice at the end. And I'm very happy with the final result. I think it will be a publishable result, which is very nice. I think this is a potential for publication, the work that Noam will be presenting today. I think Noam is not the shy type. Agree. But I think with time, Noam learned to be sort of to learn a bit what science is about and how to be a bit careful about science, and I think I saw a very strong learning curve across the last year and a half or so. So with this happy note, let's move to your presentation. Thank you.
תודה, דורי. תודה. Hello. Close the door real quick. Feel free to go out and get cookies if you need. Yeah, thank you for coming. I really appreciate it, really. Dad is here, which is really nice. He's usually in California. He didn't come just for me, don't worry. But yeah, this is my final kind of, this is a summation of my master's thesis. It's been really nice for me. As Dory said, I came from a computer science background. My first degree is in computer science. I got it at UC Santa Cruz. So biology was a whole different world for me. It was a really kind of big change, and I learned a lot. And this faculty was really great to be in, really supportive, really open to my incessant, annoying questions all the time. And yeah, they were very patient with me, so thank you. So my work here is called Temporal Relationships in Graded Good Cell Networks. This was done in the Spatial Perception and Memory Lab with Derdikman. Yeah, and let's begin. Okay, so had to do the hexagon grid thing. It's relevant, I promise. Okay, so this is the overview of the presentation I'm going to give. Basically, what we are concerned about in the lab, amongst other things, is how do mammals compute navigation? Mammals can navigate. We know this. Dogs, cats, rats, they all get around. They all manage to find their way home. And in the lab, we want to know, how does this work? We're looking at the neurons. We want to see how the neurons are networked, the activity of the networks. And specifically in this research, we were looking at grid cell networks, and we're kind of looking at the features of the network. And in order to answer this question, we analyzed data from the Moser lab, which is a very important lab Dory has very close ties with. And we looked at their data of neuron activity recorded in the entorhinal cortex during hippocampal inactivation. And don't worry, I'm going to get all into that. Don't fall asleep yet. Yeah, okay, so let's first start with a little bit of anatomy. Like I said, I'm from computer science. Anatomy is not my strong point, so I've made it very, very simple for myself and hopefully for people less familiar with it. So basically, we're looking at the hippocampal formation. This includes the hippocampus and the entorhinal cortex. This part of the brain is super old. It's very, very kind of evolutionary old, and it's conserved across all mammals. And basically, when you see something like that in the brain that has been conserved for so many millions of years, we know that it's really important. And here we see it in a human brain and also in the rat brain. And we know from previous work on human patients and animals that this is really, really important for also navigation, which is kind of what we're dealing with here. Okay, so because not everybody here is familiar with how we collect data here, I'm going to really kind of briefly go through how we collect the data. So really, data collection starts with this little piece of electronics called a tetrode. This is a device that has very, very small wires. Like smaller than the width of a hair. They're in microns. They're very tiny, and in the lab, apparently they're very hard to work with. And we implant this into the rat brain. In our case, this we didn't do this. Like I said, this was in the Moser lab, but we do similar stuff in our lab. So in this case, this would be in the entorhinal cortex. And these small wires, they measure very small changes in voltage. Essentially, we're measuring the voltage not inside the cell, but outside the cell. And we do this as we let a rat explore an arena. So basically, the rat is in some kind of enclosure, it's moving around. While it's moving around, we record these voltages. And what we're trying to find in the voltages are action potentials. Action potentials are really the language of the brain, you know, the sort of maybe more or less consensus on that. And so we try to look in this voltage to find these action potentials and essentially when the neurons are firing. So we find this data, find where these action potentials are, where these spikes in the voltage are. We combine this information with the video of the rat's location, and combining this information together, we get a kind of map showing us where in space the cell was firing. And this is really the data that we're working with here. You know, so we're getting kind of, how much the cell is firing in a particular location. Okay, so there are many of these spatially selective cells. Let's start with the hippocampus. Hippocampus has very famous place cells. Place cells were a very big discovery in the 1970s. John O'Keefe was digging around in the hippocampus, and he happened to find these cells. And they're amazing. As the rat moves around, the rat moves all over, and these cells only fire in a very specific location. And it's very kind of repeatable, very consistent. And this was a very big discovery in the 1970s. A couple decades later, in the 90s and 2000s, people were exploring the entorhinal cortex, and they found more of these spatially selective cells. In the 90s, Jeffrey Taub found head-direction cells, and these cells are tuned to direction. So basically, they only fire when the rat is facing a particular direction. This was also a big discovery. There's also speed cells in the entorhinal cortex. These are cells that respond to velocity. They fire more as the rat is moving faster, and also border cells, which kind of fire along the walls of the enclosure. So basically, if the rat's around here, then the cell only fires along this border. And in 2005, in the Moser lab, they discovered grid cells. And that's really what we're concerned with. And so let's talk a little bit more about grid cells. So like I said, they were discovered in 2005 in the Moser lab. This was a big discovery. It led to a Nobel Prize, a holy grail of science. I need to say that it was won along with the discovery of the place cell. It was kind of a combined Nobel, but still, big deal, not to lessen their work. But yeah, these cells are really, really exceptional. And so I want to show you a little bit how they look like when you're recording them in the lab. Yeah, so this is a video that Dory made. Dory worked in this lab, did a postdoc there, and he made this. And you can see as the rat moves around the arena, what you're seeing on the screen is where a particular cell that's being recorded is firing. And as the rat moves around, you see that there's kind of a pattern. It's not totally random. The cell is definitely not firing everywhere. And you can see that there's kind of clusters where the cell is firing. And some cells are clearer than others, but essentially, if you look at many of them, you see that they're firing in a very regular way. And what we do in the lab, and other people who study good cells do, is try to quantify the regularity of this pattern. So one of the ways we do that is something called the grid score. I'm going to just show you really briefly how we calculate this. First thing we do, we take a map of where the neurons spike. We convert this into a rate map, kind of saying how much the neurons spiked in that spot. If it spiked a lot, then it's going to get a brighter color. If it didn't spike so much, it's going to get a darker color. And then what we do with the same map is do an autocorrelation. An autocorrelation is a really interesting kind of mathematical transformation that was new for me. And it's really nice. It kind of measures the periodicity of this pattern. And when you do it in two-dimensional space, you can kind of see in which directions the pattern repeats itself. So this kind of tells us that, of course, when you lay the cell on top of itself, the pattern is going to line up perfectly. And when you and then the other kind of areas where there's higher, brighter color, is directions in which the pattern repeats itself also. So there's 6 of them. And what this is kind of telling us is that this pattern repeats itself in kind of a hexagonal pattern, essentially. And this is what we use to get the grid score.
Ask a question. The original grid cell depends on the boundary condition in if the square or circle.
Yeah, that can make a difference. Circle and square, not so much, but there are things that you can do to kind of change the shape of the grid cell. Like say you recorded in this square environment, and you got like this shape, and then you move this wall over, like you stretch the wall, you would see that many times these entire grid patterns just kind of stretch. And this is like super interesting, and there's all kinds of mathematical models of how this happens. And maybe I'll even mention it later in the presentation. But yeah, so the boundaries can make a difference. So, how is this pattern computed? So many perfect triangles, so geometrical. It's really amazing in nature to find something that's so kind of mathy and so regular, and it really kind of begs the question: how is the brain generating this? How is it doing this math? And so it's a mathy question. So we have kind of mathematical approaches to this, and one thing we do is kind of make models of these neural networks and try to see if we can recreate this computation. There's 2 major classes of models, in my opinion. And the first, I would say, is a single cell model. Essentially, this is a mathematical model where one cell is doing the entire computation. This is one example here, the adaptation model by Kaufman-Tevers. And in this model, they get all their information from place cells, and that allows them to generate the grid. Now, another model is the network model. And in this model, these grid cells actually communicate with each other. And this communication and the connectivity between the cells is what enables the cells to produce this grid. I just got back from a computational neuroscience workshop, and the impression I got there is the network model is maybe a little bit holding stronger in the field, but it's not a dead-end story. It hasn't been.
The jury's still out.
The jury's still out, yes. So that's a big question for us. The jury's still out, and this is another thing we can do with the science is help support, the idea of the model. Okay, so I'm gonna just give an example of the Tractor manifold model. This is kind of first described by Fuchs and Turetsky in 2006. In this model, basically, you have a sheet, and every pixel on this sheet is a neuron. And what you have on top of this sheet of neurons is a pattern. And the kind of bright spots are this pattern. And basically the connectivity is kind of hat-shaped. That's why I put up the hat. I don't want to say Mexican hat. I think that's a little passe, I don't know. But let's say a hat-shaped connectivity matrix. And essentially that creates this this firing pattern here of these kind of...
So you create the head shape by the inhibition surrounding the main excitatory connectivity pattern?
Yeah. I didn't really describe that well, but yeah. Essentially, you're creating a network model, and in this model you can have excitatory connections and inhibitory connections, and if you kind of shape them in a certain way, you create a certain type of activity pattern. And because they're shaped in this kind of surround, center surround thing, they create a activity pattern that looks like this. And so this activity pattern with the dots, it gets moved around by velocity. Basically, the cells receive velocity as input, and that moves this pattern kind of up and down, side to side, in all the directions, depending on the rat's velocity. And what happens, like I said, every pixel is a neuron. And so you see right now the pattern is over this neuron. And as the cells receive velocity input, it pushes the pattern this way. So now the pattern is off the neuron. My mouse is got over. So it's not firing, but the dots are getting closer. And eventually, another dot lines up with the neuron, and now it fires again. And so when you look at it, and you look at this particular neuron, this is the neuron in green, and you'll see when the pattern goes over the neuron, that's when the cell is going to fire. And this is
what creates the movement of the pattern?
the velocity.
Of the animal?
Yes.
As she walks?
Yeah. So the idea is the network is getting kind of input from outside the network, some kind of proprioception input or visual or whatever, and that input is what's moving the pattern around. And when the pattern goes over the neuron, that's when, if you were recording, like how we record, that's when you would see the fire. And eventually, this animation doesn't show it. I guess Gilad didn't really feel like going all the way. But eventually this would make a grid pattern. So this is just one model. This work that we did didn't necessarily go into modeling too deeply, but what we can do in this work is kind of find some evidence to support, network activity or no network activity, and that's kind of how I'm relating this to the work that we did. Like I said, part of this work is kind of supporting certain models over others. So what do we know about grid cell connectivity already? We know from very good work by Cui in 2013, also from the Moser lab, that grid cells are connected by inhibitory neurons, interneurons. So they are connected. There is some level of connectivity, there is some level of network, at least at the connectivity level. We know that grid cells receive input from other regions.
Wait a minute. Is this cortex different from any other cortexes?
Yes.
Why shouldn't they be connected by inhibitory neurons? Why this is big news?
It is different.
So can you elaborate on that?
I don't think I will. I'm just going to say that they're connected.
I'll just say a quick word. It's a bit of a different cortex. So it's organized differently than the cortex you're familiar with. So basically, you have the stellate cells in layer 2, for example. So and these stellate cells do not have direct connections between them, but only via inhibitory interneurons.
Okay, so these are stellate cells, not
These are
pyramidal
basically the
cells.
stellate cells.
Well, it can be both.
But the
But they're
grid neurons
projecting
are not
stellate cells.
pyramidal neurons?
They're both.
Well,
They're
the
both.
grid cells are
Yeah.
mostly stellate cells. They're layer 2 stellate cells which project out of the layer.
Essentially, what's important, at least in respect to this project, is that there is connections between them.
But indirect.
Indirect connections, but connections nonetheless. So let's see, how much Okay, yeah. And so, yeah, this is, yeah, so a key question here is how much of the grid pattern is due to input and how much is computed elsewhere. So we see these grid patterns in the cells and we want to know, is this being calculated inside the grid cell network? Is it in the entorhinal cortex? Or maybe it's calculated somewhere else, like maybe this whole grid pattern is from some other part of the brain and all the work's done and they're just kind of showing the output. So how do we do this? We try to remove the input. We're not the first people to have this idea, and this was actually done with mice previously, and the idea was to put them in the dark, so basically these grid cells aren't getting visual input anymore because the mice can't see anything, and then we want to look, and we want to see what remains, if at all. Can they still make the grid pattern? What happens when they're in the dark? So that was done in mice, and the data that we looked at took away the input in another way, which was by inactivating the hippocampus. This was, like I said, I mentioned the study earlier. Getting it? Set up? Okay. Yeah, so yeah, this was done previously in 2013 with Bonnevie, like I mentioned earlier, in the Moser lab. So they inactivated the hippocampus. So let me go over it. This is the data we used, and this is the experiment they did. So essentially, the first thing they did was measure in the entorhinal cortex and measure cells as they're exposed in the arena, like I said earlier. And then, after doing that and finding a set of grid cells, they inactivated the hippocampus. This was done with mucimol, which is a GABA agonist. Basically, it kind of puts the cells to sleep. The activity goes down, they're not really firing, and they quantified this, kind of how much the cells went to sleep. And so after the hippocampus goes nighty-night, then they start measuring from the entorhinal cortex again in the same spot, and they're trying to measure the same cells. And
so now the hippocampus is very big, right?
yeah.
So did they map first which region goes to which region, and then they injected the mucimol?
Yeah, they injected it into CA1.
The whole CA1? The hippocampus is huge.
Yeah, I'm not too clear on that, and actually, yeah, I think from my point of view, they showed in their paper the kind of effect, the degradation of the grid cells and the connectivity. So that was kind of already published.
I'll say a word about
Yeah.
it. So they also controlled by recording. So first, mucimol is never a very clean experiment, of course. But they also looked to what extent they also recorded place cells. So they had to do, sometimes they did it simultaneously with the recording of the grid cells.
Answering what is the connectivity between the entorhinal cortex
But
and
this is known that there is a gradient, meaning dorsal is more
Right.
connected to dorsal, ventral is more connected to ventral.
so they
I
watched
guess
it beforehand.
I guess they mostly went to dorsal. I would think.
Yeah, they published our paper which essentially said that the entorhinal cortex needs excitatory input from the hippocampus in order for the grid cells to function. So they basically showed that, and we're not disagreeing with it. Seems valid. So yeah, they did show that there's some level of connectivity because you see the effect.
But at least they inactivated a large enough part of the hippocampus to cause the
yeah.
grid cells to disappear. So this at least
Yeah.
we can say.
And what's nice is that they didn't stop all the activity in the entorhinal cortex either. That was really nice. It's not like everything in the entorhinal cortex also went to sleep. There was still very lively activity in there. That was still, yeah.
But I wanted to ask, the GABA agonist, it inactivates the hippocampus or just affects its work?
Yeah, GABA is neurons are kind of, I'm less good on this, but yeah, they're all over the brain, probably, I think in the body also. They are inhibitory neurons. So basically, if, there's a neuron that's connected to one of these GABA neurons, if the GABA neuron fires, it inhibits this neuron from firing. So basically, any cell that it's connected to is going to fire less. And so by using an agonist as opposed to an antagonist, you make these inhibitory neurons fire more, which makes everything around it fire less. So, yeah, it's used a lot in medicine. It's like a general sedative. I actually read that this muscimol was actually found in mushrooms, in actually the psychedelic mushrooms, but it's not the psychedelic part. It's a different part. It's like a sedative part.
So did they infuse the muscimol constantly, or just one shot injection?
I think it was one injection, but Dory knows better than me. It was
One shot.
One shot, yeah. But yeah, looking at the time, yeah, Jackie, that's like a relevant question, yeah. And because of that, we only looked in like the first 45 minutes of the recording, because eventually it does wear off. Yeah. Okay, so measure the same cells again with the hippocampus inactivated. And again, the tetrode has stayed in the same spot the whole time, so you're assuming you're measuring the same cells. But just to double check that you are measuring the same cells, you measure a third time after the muscimol wears off, and you kind of see and you hope that the activity is similar before the muscimol and after. And you want to see that you didn't cause any permanent brain damage, or the tetrode moved and you're measuring a different cell. That's really important. Okay, so all in all, they recorded 41 groups, 301 cells total. It's really hard to do all this recording, and it was very impressive. This is part of why they won the Nobel, because this is really hard. Really nice data to work with. So yeah, we got to look at this data and analyze it, and so I want to show you a little bit of what it looked like. Yeah, so basically we were quantifying the grid score and the gridness of these cells before the hippocampus, kind of like I showed you how you calculate the grid score with the autocorrelation. This is during, so you can see the pattern doesn't look so great, and after. And you can see that, you know, in this example, the pattern went back, and this was for the majority of the cells in this group. It was really nice work, very good reporting. And again, this is from the entorhinal cortex, while the hippocampus is moving.
No,
Yeah.
can you comment about the firing rates? Not the location where they fire, but the firing rates of the neurons in the pre, during, and post?
Yeah, so they said in their paper in 2013 that overall the rates went down, but there was still plenty of activity. It wasn't like the rates totally went away. But I think
20% or 50% or 70%?
Yeah, that's a good question. I don't think I can answer that. Yeah. But I know from analyzing the data there was plenty of activity. Like there wasn't a shortage of, like in my data, I was dealing with like 5,000 spikes over, 45 minutes.
During, and in the pre, how many spikes are there?
On the scale.
Similar number.
Yeah, so like there was enough activity too. But yeah, I think overall it did go down some amount. Yeah, from their study. Okay, yeah. I just want to point out here that you can already see that during this inactivation of the hippocampus, balagan, chaos. Look at these patterns. They're all a mess. No more grid. It's just total madness.
You don't do a complete balagan, because it looks like there's some board suppression there in all the cells for some reason.
Okay,
On the
so
right,
yeah.
I don't know. Did you analyze that?
We did analyze the balagan, and we did find some order in the chaos. So there actually wasn't a total balagan. And this project was digging through the balagan. But spatially, it sort of looks like a balagan.
Yeah, less order.
Less order. Unless you're really good like Yoram and you can actually see it, then it looks like balagan. So our analysis. Okay. So we were mostly concerned with grid cells where the grid disappeared. So some of these grid cells were not affected by the muscimol, who knows why, but we were really concerned with the ones that were affected. And this was about, maybe like a quarter of them, quarter of the grid cell, probably more. But of all the cells recorded, which not all of them were grid cells, a quarter of those at least did have their grid scores significantly degraded. This was the thresholds that we used. We had a minimum grid score of 0.3. We wanted ones that decreased by at least 0.2 during the inactivation. So overall, we ended up with about 70 cells over 17 groups. And we organized them in pairs of
So just a question
yeah.
about the degradation. So in the same animal, when you saw a loss of grid cells, it was across the board, or so I'm trying. Was it salt and pepper? So some in the same animal, same experiment, you could find some with good grid cells and some not. And what was the spatial distances between them?
That's a good
Or
question.
was it just some experiments were good and some
it was
were
more
bad?
like that. It was more like that. It's not something I looked at super explicitly, but from what I remember about looking through the data a bunch, it was more like that. Like on some days when they recorded, it seemed like all of the good cells remained, and on the day, so it was kind of less.
Pointing to some technical issues of injecting,
Yeah.
which is okay.
Nobel winners can make mistakes.
It's not mistake.
Yeah. Okay.
Such is life. We inject constantly.
Yeah, yes. And so, yeah, so really the question that we wanted to ask was, does anything remain? Does anything remain in the mess? You know, the spatial kind of pattern, the spatial activity looks like it's really deteriorating, but maybe there's other stuff that does remain. Maybe there is some kind of activity. Okay. Oh, I almost skipped the GUI. So now we're going to move to the GUI. And this is, yeah, so this was, okay, something, okay, so now I'm going to show you grid cell activity. So this is playing back this is playing back the Bonnevie data. Maybe we can make it darker in here. Yeah. So this is the data, and this is just basically replaying data back. Yeah, this is, yeah, maybe I will take a second. This is something that I put together in MATLAB, the kind of GUI function. And I just wanted to say, for anybody here who's doing research,